Okay, so as Dr. Nabob said, we're going to talk about the hope and hype of stem cell therapy to treat heart disease. And I want you to sit back and I'm going to tell you a story about how stem cells have come about. And I'm going to give you several examples on how the hype of stem cell therapy, even outside treatment for the heart disease, has harmed the society. I have no disclosures, and I want to start by defining life expectancy. Life expectancy is defined as a statistical measure of the average time an organism is expected to live based on the year of their birth, their current age, and other demographic factors, including sex. And if we look at throughout the history, the life expectancy since the early 1900s has been steadily going up. It was in 1917 because of the pandemic, the Spanish flu pandemic, there was a significant drop, but it continues to go up. And this is the chart that indicates what's the probability of 65-year-old male surviving to different ages. And as you can see, there's a 50% chance that that a 65-year-old male will live up beyond the age of 87 to 88 years old. So the question is, since the life expectancy in the US has steadily been going up, except for the last year that there was a drop, currently the life expectancy in this country is about 77.3 years old, about 80 years old for women, and about a little over 75 years old for male. The question that remains is, if the medical advancement continues, will there be a linear increase in life expectancy throughout the years to come? Or if we just get to the bottom of this, if someone asks the question, what happens to life expectancy if we eliminate all cancers, or if we eliminate all heart disease or the disease of respiratory system? What happens to life expectancy? And to my surprise, when I started just researching this area, to my surprise, this has been a very active area of research by several scientists. In fact, in the 1970s, Tibor Paradox was published, and it said that even though a particular disease may represent a large portion of all deaths, its elimination does not assure huge gains in life expectancy. On the contrary, it may result in only modest life expectancy increases. And this is a phenomenon that's called Tibor Paradox. So if I told you that starting tomorrow, all heart diseases will be eliminated, what do you think the life expectancy will change? So to my surprise, when I did search on this, if we eliminated all cancer, life expectancy would only increase by 3.5 years. And if we eliminated all cardiovascular disease, life expectancy or potential gains in life expectancy would increase by 3.1 years. And this is because as we eliminate one cause of mortality and morbidity, we're just placing it into another category. The longest living individual that has been documented is a French woman, Jean-Louis Coleman, and she was born in 1875 and she lived to 1997. That was a total of 122 years and 164 days. She had several family members who lived to their 90s and 100s. She rode her bicycle until her 100th birthday, and she was neither athletic nor fanatical about her health. She drank wine daily and consumed more than 1 kilogram of chocolate weekly. And she actually goes on to talk about how she met Van Gogh when she was a little child, and when she went to his shop and bought some paint. And very interestingly, she smoked from the age of 21 to 117. And this is very interesting that we see people, as physicians, we see people who smoke and are not athletic, don't care about their health, and they have a number of different diseases that they present with in their early decades of life. But this individual who smoked and was not very careful about her health and her daily activities, she lived to be 122 years old. So what is it about people who live to different ages and they present differently? For instance, this individual is only 50 years old and she is, or he is 80 years old, and you can see the difference between the features, their presentation. So there is a difference between the numerical age and biological age. So just because someone is 60 years old doesn't mean that their biological age is also 60. And this is an active area of research where people are coming up with the difference between biological age and numerical age. So the longevity and tissue aging, the traditional view has been looking at two areas. One, intrinsic factors that affect the aging, and one, extrinsic factors that affect aging. For intrinsic factors include what we're born with, our genes, the mutations. However, extrinsic related factors are what we're exposed to, and those are environmental stresses. We're exposed to infections, injury, and the stress that we encounter. And the thought has been that in combination, both intrinsic and extrinsic related factors cause molecular changes that lead to tissue aging. However, there has been a new thought that this is not as simple as initially thought. And there are tissue stem cells that may play an important factor in terms of developing or leading to tissue aging. And the importance of stem cells in aging and longevity is starting to be appreciated. And this is what causes it's this tissue stem cells that cause changes in molecular aging and also tissue aging. So what exactly is a stem cell? If you don't take anything from this talk, I just want you to understand a definition of stem cell. Many people talk about stem cells. Many people claim to be experts in stem cells, but they probably don't know the basic definition of stem cell. A stem cell has to have two properties. One is the ability to self-renew, and the other is to be able to differentiate into many cells. These are both characteristics of a stem cell. And many people who claim that they are talking about stem cells, in fact, they're not relating. The type of cells that they're talking about is not actually stem cell because they don't possess these two characteristics. So, for example, a single cell that's considered a stem cell can replicate, can renew itself, can expand, and make many more cells that are similar to itself, and they can also differentiate into different progenies and generate different cell types. For example, making neurons, making heart cells, so on and so forth. So here we have, if you look at embryonic stem cells, these are stem cells that are generated or that are taken from the fertilized egg at the early stages of the blastocyst formation. When we take the inner cell mass and we propagate them in vitro, these are called embryonic stem cells. And they are considered to be pluripotent. And what pluripotent means is it has the ability to generate all different cell types of the body, including neurons, muscle cells, cardiac cells, liver cells, any cell that you can think of, it's capable of generating them. And by generating these different cell types, we have the ability to provide therapeutic options for cell transplantation. However, what I've told you so far has been mostly theoretic, and we have not gotten to a point of harvesting or applying the therapeutic potential of stem cells in all these different fields. So I want to tell you a brief history of stem cells and how it came about. So the first cloning happened in 1938 when Hans Spann published his first work on nuclear transfer using salamander embryos. And this was the first example of cloning. It was in 1978 that Louis Brown was born as a first baby from in vitro fertilization. And since 1978, it's been estimated that more than 5 million births have been aided by IVF. And that is more than 1% of all live births. And this continues to increase. IVF has become increasingly popular among aging couples, and also couples who are not capable of conceiving naturally. In 1981, Sir Martin Evans and Matthew Kaufman derived pluripotent stem cells from mouse embryos. And pluripotent stem cells, as I said, are the cells that are derived from the fertilized egg, and in this case was a mouse. And these cells have the ability to generate all different cell types. And in 1988, the first adult stem cell was discovered by Irv Weisman, who I had the opportunity to work with for 5 years at Stanford before coming to UCLA. And the hematopoietic or blood-forming stem cells is the only example of adult tissue stem cell that has been shown to have any therapeutic relevance. And we'll talk about this more. In 1995, the NIH Human Embryo Research Panel recommended made a recommendation to federal government to fund research using both embryos left over from IVF and embryos created for experimentation. And this was a huge recommendation that did not go into effect. Immediately after that, a divided government and Congress intervened and passed the Diki Amendment. And this Diki Amendment has been passed on every administration since 1995. And it said that it prohibited NIH or federal funding for creation of human embryos for research purposes, or research in which a human embryo is destroyed, discarded, or knowingly subjected to risk of injury or death. So meaning you can only use what has been left over by IVF, you can't generate anymore. So it made it illegal for government to fund, but not for private citizens to carry out these kind of research. In 1997, Dolly the sheep was the first mammal that was cloned. And in 1998, Jimmy Thompson from University of Wisconsin isolated human embryonic stem cells. These are the pluripotent stem cells equivalent to what was previously discovered from the mouse embryos. In 1999, the President's National Bioethics Advisory Commission again recommended that human embryonic stem cells harvested from embryos discarded after IVF eligible for federal funding, but Diki Amendment stands in this way. And Harriet Robb, which was the chief counsel for Department of Human and Health Services, wrote a piece that became a very, very influential note that government cannot fund any destruction of embryos, but can fund the research once the stem cells are created, meaning that whatever IVF makes and they're not using it can be used for research. And President Bush took 7 months to issue his decision regarding stem cell research in a TV speech. And he stated that the derivation process, what had to be initiated prior to August of 2001, they must have been derived from embryos created for reproductive purposes and no longer needed, and informed consent must have been obtained. And this was seen as a compromise and was heavily criticized by the scientific community. And very interestingly, initially thought that there were 77 lines available worldwide, but turned out there were only 21 human embryonic stem cell lines available. And some of these lines were contaminated and others had genetic mutations. So it put a lot of money and power in the hands of those that own these lines. So during the Bush era, the states spoke out. In 2004, California Proposition 71 authorized the state to fund $3 billion on stem cell research. It was making it larger, a larger funder than the federal government. And you may know that this last year, California proposition was passed again for another $5 billion in this area. And also other states followed, and in 2005 poll, 70% of Americans favored loosening the Bush administration restrictions, including more than 50% of conservatives. In 2007, human skins were reprogrammed to pluripotent stem cells by Yamanaka and Thompson, and this overcame some of the previous restrictions on using embryonic stem cells. And Obama campaigned to increase federal funding for stem cell research, but unfortunately, the Diki Amendment stood in the way, and he was unable to push it forward. And in the years between Clinton and Obama, many other stem cell lines were created by private companies. And as many of you know, in 2012, Gordon and Yamanaka won the Nobel Prize in Medicine for their groundbreaking stem cell research. So this is just a brief history of stem cell, highlighting the importance of this field, not only in the scientific arena, but also the extent of political involvement in this. This has led to a hype of stem cells, and this is in the media all the time, that there are many companies that are created and many companies that are promoting use of stem cells. There's even a book, A Miracle of Stem Cell in Heart Repair, and we see it on in the media all the time that celebrities are getting stem cell treatment. But if we look at the stem cell therapy sales, it continues to go up over the years. However, if we look at the net worth of these companies, they're always going down. So there's something going on that doesn't match up. So I want to tell you a story about stem cell, use of stem cell. So in 2005, on August 15th of 2005, a 75-year-old Hawaiian singer, some of you may know Don Ho, who's also known as King of Waikiki, underwent an experimental procedure in Thailand, where his bone marrow was injected directly into his failing heart. He was expected to live for 3 years on his usual medical regimen because of his heart failure. But he wanted to do this experimental procedure to see if he could get any benefit from it. So he booked a stem cell treatment package that included round-trip tickets to Bangkok, a 5-star hotel, and a VIP hospital room. So his bone marrow was aspirated and was sent to Israel. He himself flew from Hawaii to Thailand. His interventional cardiologist went from Miami to Thailand to perform this procedure. And his processed bone marrow came from Israel all the way to Thailand for this procedure to be done. The total cost was, this is 2005, over $130,000. And the USA Today headline the next day said, "Big stem cell success story." He was interviewed and he said, "I have more and more energy. I can feel my heart pumping stronger. I'm ready to go another 50 years with my new heart." This was the day after he received the quote-unquote stem cell therapy. However, he died of heart failure 6 months later, and the cardiologist who performed the procedure lost his license in 2010 because of performing a procedure that was not scientifically proven in a patient by taking advantage of a patient who is vulnerable to these kind of predatory procedures. So medical tourism, and specifically stem cell tourism, has become a huge industry. It's more than a $60 billion per year industry. And about a decade ago, more than 500,000 Americans traveled outside of the US for certain kind of care. And this can be seen by the cost. For example, a bypass surgery in the US that could cost approximately $150,000 could cost about $5,000 in India. However, there are other unproven therapies that have also been advertised, and many people are getting lured into performing these procedures without any efficacy. The for-profit clinics offering stem cell therapy, and I put this stem cell therapy in code because to me it means there is no meaning to it, have been on the rise, and there are more than 1,300 clinics nationwide. In California alone, there are more than 200 clinics for stem cells. And Beverly Hills is a hotspot for consumer stem cell clinics, with at least 8 clinics located near a 1-mile stretch of Wilshire Boulevard. And these treatments are not approved by the FDA. And I call them snake oil. And this is something that absolutely has no scientific proof for being efficacious in treating any kind of medical therapy. As I mentioned before, the only stem cell that has been shown, the only treatment from stem cells that has been proven for medical efficacy is the use of bone marrow cells for certain cancers, certain blood cancers. Other people who claim that they're going to take stem cells and injected into knees and hips and injected into arteries for improving heart disease, these are not proven stem cell therapies. There's absolutely no scientific basis behind this. There's a difference between a clinical trial that's being done by a reputable academic center versus a clinical procedure that's done by a clinician in one clinic. There's a significant difference between them because one is based on scientific finding from a large population. The other one comes from the personal opinion and an individual's experience. So this is always an important point to make to patients who are vulnerable and do not know the potential harm that these could cause. A while ago, I had a patient who came to CCU under my care who had received stem cell for pulmonary hypertension and had developed these blood clots in her lungs. And this was an unproven therapy that was performed outside the US. And unfortunately, this patient was lured into it. So while we talk about the stem cell and how it's important, how the ethics are so important, I wanted to give you an example of how cloning, a scientific breakthrough in the field of molecular biology, underwent the same trials and error, trials and tribulations in the 1970s, and it came out victorious. And this was the effort of a molecular biologist by name of Paul Berg at Stanford. And he was a leading scientist in the field of recombinant DNA. And he organized the Asilomar Conference. And the Asilomar Conference is very, very well known. If you Google Asilomar Conference, you can actually read about it and find out what actually happened during that conference. He organized this conference because his fellow scientists were concerned about the type of work that he was doing and the potential biohazard that could come out of it. So what exactly is recombinant DNA and cloning? In a nutshell, recombinant DNA and cloning is taking a piece of DNA from a different organism and putting it into the host of another organism, into the nucleus of another organism. And by ligating it into the host DNA, that piece of gene becomes part of the host and can be translated and transcribed. So, for example, you can take a gene that codes for certain protein and you can put it into another organism so that protein can be made by this individual, by this different organism. This was several days. The Asilomar Conference was several days, and it was not only for scientists. It brought in lawyers, it brought in federal government and public individuals, and the collective effort of these people led to certain rules and regulations by avoiding the limits and restrictions of government legislation so that they could go on and use this technology, use this significant scientific advancement. Because it wasn't long after this Asilomar conference that the same technology, cloning, was used to take the human gene for insulin and put it into a bacteria so we can make insulin. And this is the basis of how diabetes is treated by insulin. We're making insulin in a bacteria by taking the human gene and putting it into the bacteria, so the bacteria goes on to generate the protein for insulin. And the insulin is isolated from the bacteria because the gene is human, the protein is also compatible with human, and it can be used. So this is an example of how scientists, the public and government can come together and overcome certain limitations in scientific advancement. But has that been the case all throughout? I want to give you another example. And as many of you may have heard about CRISPR-Cas9 technologies. This is one of the greatest advancements in the field of science and medicine over the past decade. CRISPR, or it stands for Clustered Regulatory and Interspaced Short Palindromic Repeat. And these are sequences that were initially discovered in a bacterial genome as an adaptive immune system. And Cas9 is a protein, is an enzyme that uses the CRISPR sequence as a guide to recognize and cleave specific parts of the DNA. So the way you can think about it is that this CRISPR-Cas9 can go into your genome and it can identify the area of interest, and think of it as having two pairs of scissors on each side. And you can cut out the area that you don't want, and you can actually put in a new piece of DNA in there. This is called genome edit. It's a type of genetic engineering in which DNA is inserted, deleted, or replaced using engineered nucleus or molecular scissors. And this is this genetic or genome editing is the backbone of what has led to significant scientific breakthroughs over the past decade. It was actually CRISPR-Cas9 technology was considered the breakthrough of the American Association for Advancement of Science in 2015. However, there are bioethical concerns about CRISPR-Cas9 technology. And you may have thought about it already yourself, that with this technology, you can take anything, you can take any genome of your interest, any parts of the gene of your interest, and replace it with what you would like. For example, so I want to first talk about the difference between somatic and germline. Somatic are the cells that we have right now. These don't have the ability to pass on to the next generation. Germline cells are the sperm and egg that can pass on the genes from generation to generation. So what if CRISPR-Cas9 technology is used in the germline cells? For example, what if a couple is interested in having their child having only blue eyes, and they take the germline cells and they change the gene for whatever they're interested in, blue eyes, changing the height of the individual, changing their IQ, all these things. So there's a gray line that would raise concern about the ethical issues. So this is something that I've thought about, and I'm sure that now that you're listening to this, you may think about what are the bioethics concerns about CRISPR for germline editing? And this actually happened. So harnessing an application of genome editing, although it can be significant, it could lead to disease modeling and generation of transgenic models. We can put human genes into animal models to study them. It has therapeutics effect for a certain number of diseases, including beta thalassemia, sickle cell disease, hemophilia, cystic fibrosis, Duchenne muscular dystrophy, Huntington's, so on, and treatment of infections. In 2016, the first CRISPR-Cas9 cancer clinical trial was underway. And it can knock down or activate certain genes that we want. But what if this technology is used in germline? And as I said, some of you may know what happened in 2018. In 2018, the second International Summit on Human Genome and Editing was underway in November of 2018 in Hong Kong, in collaboration with the Academy of Sciences of Hong Kong and also National Academy of Medicine of the US. And to everyone's surprise, there was a Chinese scientist by the name of He Jiankui from the Southern University of Science and Technology in Shenzhen, China. And he reported, to everyone's surprise, that he used CRISPR-Cas9 technology to alter the embryonic genes of twin girls born several days earlier. So he came to that conference, he had a talk, and to everybody's surprise, he gave a talk later that evening his talk was um, delivered again, and it was considered to be one of the highest audiences for a talk. More than 1 million people tuned in to listen to his talk because it took the world by surprise. So what happened was that in October of 2015, a couple, he started doing experimentation on a couple. The father, it was a couple, husband and wife, and the husband was HIV positive, and he wanted to do an experimentation to make sure that the offspring of this couple were HIV negative. We're going to go into it, but this study was self-funded and performed in secrecy from the hospital scientific community and was not published. The Southern University of Science and Technology in China was unaware of this. There were more than 100 scientists condemned the work immediately, and in an open letter considered it dangerous and unjustified that led to opening of the Pandora's box. But in an interview that night, he actually left the conference without notifying anyone, and he actually left his hotel without even taking his belongings because it was such a huge media attention. He received such a huge media attention that he wasn't expecting for it. But he did mention that he felt proud for being the first to do this. For what exactly did he do? So IVF embryos from healthy mother and HIV father, as I mentioned to you. So the couple wanted to have children, but they were afraid of their children to have HIV. So just as a background, the entry of the virus, the HIV virus, into target cells depends on the presence of two surface proteins, CD4, which serves as the main receptor, and either a CCR4 or a CXCR4 as a co-receptor. There are a limited number of people worldwide who harbor a mutation in the CCR5 gene, leading to expression of a truncated gene. So it provides resistance to HIV infection. So these individuals who have this mutation are resistant to HIV infection because the HIV virus is unable to be taken into the cell. You may have heard about the Berlin patient who was HIV negative, and that individual actually had a CCR5 mutation. So he used genome editing to create a specific mutation in CCR5 gene that confers innate resistance to HIV in the sperm and the egg of the mother, the sperm of the father and the egg of the mother. So what exactly happened? He took the sperm, he washed it, and he did IVF, in vitro fertilization. And when the cells started growing in a blastocyst stage, he took out a number of cells and he genetically engineered the genes and he put it back into the fertilized egg. So the twin girls were born in mid-October of 2018. We don't know about their whereabouts or their identity. They're referred to as Lulu and Nana. And we know that they're still alive. As far as Dr. He Jiankui is concerned, he was fined over $500,000 and he was imprisoned in 2019, in December of 2019, after being under house arrest for a year. And we know that the twins are alive, but we don't know what the future manifestation of their gene editing will be. One example is that we know that CCR5 mutation also causes resistance to stroke and maybe brain changes in the brain. So we don't know exactly what the future implications will be. So everyone will have their own ideas about this, and I'm not here to talk about the ethical or the moral issues of this. I have my own opinion that this was premature and it should not have been done, and this is not ethically the right stage of using this technology. We don't know if they caused any chimerism, meaning that some of the cells may have it, some of the cells may not have it. And we don't know if there's any off-target effect of CRISPR-Cas9 technology. Some of these genes may have been inadvertently inserted into other areas, and we don't know what's going to happen to those. But the question that remains is, can this technology be used for other things? For example, people who have parents who are carriers of cystic fibrosis. There is a single point mutation that causes cystic fibrosis, and it's the chances of their offsprings, their child, to have cystic fibrosis is extremely high. Can we use this technology to correct a disease like cystic fibrosis and give hope to couples who are carriers for this disease? So this is an area that is not, as far as I'm concerned, it's not black and white. There are a lot of gray areas, and people will have their own opinion about it. And I hope that the future will be in the direction of scientific advancement. And as we headed with the Asilomar conference, something like that will happen with genome editing before it goes out of hand. So we've talked about this, but what's the relevance to stem cell therapy for the heart? So going back to cardiovascular disease, as many of you know, it's the leading cause of morbidity and mortality worldwide. It's been the #1 killer, and despite advances in medical therapy and surgical therapy for acute myocardial infarction, the 1-year mortality is 13%, and 5-year prognosis for patients with heart failure is 50%, meaning 50% of people who have heart failure, who are diagnosed with heart failure, 5 years later will not be alive. And there are about 400,000 new cases of congestive heart failure that occur each year, and there are more than 2,600 deaths that occur each day from cardiovascular disease. That's 1 death over every 30 seconds. So over the course of this talk, there have been over 100 people who have died from cardiovascular disease. Unfortunately, the heart does not possess a regenerative potential. For example, you get a scar in your skin, over a few days, it's replaced by normal skin because the skin has the regenerative capacity, but the heart doesn't. In lower organisms, for example, the zebrafish and amphibians, the regenerative potential is significant. Even legs and arms can be amputated and they can be regenerated. In an adult zebrafish, about 30% of the heart can be resected, and it has the ability to regenerate itself. In the mouse, we know that during neonatal period, there is a regenerative capacity of the heart, but as it gets older, this regenerative capacity is gone. And in humans, we know that damage to the heart leads to loss of myocytes and scar formation, and hence heart failure and remodeling. So the first human heart transplantation, so because we have only limited therapies for cardiac regeneration, people had thought about doing heart transplantation. And the first one was done in December of 1967 in South Africa by Christian Bernard, a cardiac surgeon trained at Stanford. And there was a family who lost a daughter. They were going to a birthday party. They parked the car. The daughter and mother get out of the car to cross the street, and the mother and daughter were hit by the car. The daughter died, who was a teenager, and her heart was used as a first transplant. And very interestingly, the picture of the woman was actually on the front page news. This is something that we never do these days. The identity of the donors are confidential. But the heart transplant did not become an accepted practice due to high mortality from rejection. It was considered a medical curiosity, but not practical to treat patients. Because of the rejection, it didn't take off. But the scientific story is remarkable because in 1970, Swiss scientists examined soil for antifungal agents, and they looked at soil from different places, including from Wisconsin, and they discovered the compound called 24-556 that had antifungal activity. But the spectrum was very narrow and the action was very weak. So they stopped their investigation of the compound, of this compound. And they just shelved it. However, years later, through a screening process, this compound was found to reduce rejection in mice by over 1,000-fold. And this is the beginning of cyclosporin, the medication that is used to prevent rejection after organ transplantation. So if we look at heart transplantation throughout the years, cyclosporin was discovered in 1983, and this is where we are. And immediately after that, it went up. And it went up and it plateaued. And if you want to know what happened here, that it plateaued because it was just going up, this is around the time that there was at least in the US, there was a federal law for motorcyclists to wear a helmet. The majority of the donors were coming from motorcyclists. And this is the time that it was mandatory to wear a helmet. So the number of donors decreased at this time. So we do about 3,000 heart transplantation in adults in this country every year. So the adult heart was once thought as a post-mitotic and terminally differentiated organ. However, this was brought into, this was challenged by certain findings that after a heart attack, people had seen very low level of cardiomyocytes undergoing division. And there was another interesting finding, which was very controversial and was not able to be replicated, that that males receiving female donor hearts, so a male heart transplant recipient of a female heart, at the time that the patient died with their transplanted heart, they found that there were some Y chromosome, a male chromosome in their heart, meaning that there may have been some extra cardiac source of cardiomyocytes. But this turned out to be not true. So there was a very interesting study that was done in 2009 by a Swedish group that looked at the age of cardiomyocytes, and they showed that there is very, very little amount of cardiomyocyte division in humans. It's about 1% by the age of 25 and 0.45% turnover at the age of 7. However, this was an indirect way because they looked at carbon-14 date mapping of cardiomyocytes. And we wanted to do a direct way of doing this. So we generated a mouse, using technology similar to CRISPR-Cas9 technology. We generated what we call the rainbow mouse by putting different fluorescent proteins into the genome so that we can turn every cardiomyocyte into a different color. And these are hearts of different mice when we made these rainbow mice. And we showed that there are areas during development, there are areas of the same color cardiomyocytes, indicating that they may have been generated from the same ancestor. And we actually went ahead and we actually looked at every individual heart, every individual cardiomyocyte, the muscle cells in the heart, and they have the different colors. And we showed that there are areas that could be considered as clonal, indicating that there could be a parental cell for the generation of these cardiomyocytes, as can be shown here. So we next went ahead and asked the question: what happens during injury when we induce a myocardial infarction? Can these cardiomyocytes undergo division? And we found that very interestingly, while in the early stages of development in mice, when the mouse has a heart attack, we induce a myocardial infarction in a one-day-old mouse. We see that the heart has the ability to undergo regeneration. These are the same color cardiomyocytes. However, when we do the same thing in adults, we don't see any clones of cells indicating that adults have lost their ability to regenerate their myocardium. And this is an example of what we think is a clonal hierarchical model for stem cell therapy, indicating that there are stem cells during early stages of development, and these mature to terminally differentiated cells as they mature. So after a heart attack, many people have considered cell therapy for cardiac regenerations. People have put bone marrow cells in the heart, and we've shown that this doesn't work. Skeletal myoblasts, They've put skeletal cells in the heart, and this has led to arrhythmogenicity, arrhythmias. We have shown, based on our studies, that endogenous cardiomyocyte proliferation is limited, and there aren't any resident cardiac stem cells. How about embryonic stem cells that we talked about earlier? Maybe they would be a source for cardiac regeneration. And this has been the area of research in my laboratory, where we take embryonic stem cells and we generate chamber-specific cardiomyocytes, indicating that the cardiomyocytes that are in the different areas of the heart have different properties. For example, the cardiomyocytes that are in the left ventricle may be different than the ones that are in the right ventricle. And we have used CRISPR-Cas9 technology, as I can show, as I'm showing here, to generate embryonic stem cells so that we can differentiate them and generate cardiomyocytes. And as you can see, this is embryonic stem cells that we have cultured in vitro, and they start beating, and we use these cells for cell transplantation. And we put a marker in them so that we can actually visualize them when they are transplanted. So these are beating cells that with every beat, they give off a fluorescent protein, fluorescent signal, so we can identify whether they've integrated into the heart's cardiomyocytes or not. So we have taken this approach of transplanting these cells. We have started preclinical studies of transplanting these cells in pigs, and we have done MRI and functional studies, and we have shown that we can actually identify the transplanted cells, and we have shown that they actually integrate into the heart and they mature. However, they do not form electromechanical coupling to the rest of the myocardium. And this is just an example of culturing these cells. We can use these as a model for arrhythmias, for disease modeling, and for drug testing. So before I finish this talk, I wanted to just tell you a little story about aging. I started with aging and I wanted to finish with aging. And old age has been suggested to be associated with a decline in cognitive function. And there are some studies that have suggested that there may be factors in the young blood that can restore hippocampal synaptic plasticity, improve memory and learning in aged mice. And we asked the question, can young blood reverse age-related impairment in plasticity, cognitive function, and also cardiac dysfunction? So we went ahead and performed this very interesting study of taking neonatal blood from mice. And by neonatal, I mean from 1 to 3 days old. And we induced a heart attack in an older mouse. And we gave them the neonatal blood or old blood. And this, as can be seen, this is the heart function. After a heart attack, when they receive old plasma, not much is happening, the heart fails. But when they receive neonatal plasma, they go back to near normal, meaning that there are factors in the neonatal plasma that may induce regeneration of the heart. And we showed that the reverse of that is also correct as well. When old plasma is given to young pups undergoing myocardial infarction, it prevents myocardial regeneration. So our goal is, this has been a very, very interesting project in our laboratory, that we're trying to identify factors in young serum that may actually prevent aging of the heart, and also prevent adverse remodeling of the heart after a myocardial injury. And we're in the process of identifying the factors. And we have identified several factors, and we're doing preclinical studies to determine their efficacy in preventing development of heart failure after heart attack. So with that, this is basically what I had for today. I hope I believe stem cell therapy hope is real, but has been overshadowed by hype. I hope you take the claims that you see in the media with a grain of salt. Please make sure that there is sound scientific evidence behind the claims that are made. And this reminds me of a story that 3 umpires were asked how they call balls and strikes. The first one said, I call the way I see them. So this individual acknowledges perception and reality are blurred. Another umpire said, I call them the way they are. And this person confuses perception with reality. And the third one said, they ain't nothing till I call them, and this person defines reality. And that's unfortunately how stem cell area has been plagued by bad science and also a rush into clinical application of scientifically unproven media. So I'd like to thank members of my laboratory and also the sources of funding for the work that has been done at UCLA. But most importantly, I would like to thank my wife and my 2 kids, Omid and Avid, and I would be happy to answer any questions that you may have. Thank you very much.
Thank you so much, Dr. Ardehali. I hope we can convince you to talk for us in future. It was super, super. And in other Zoom meetings and other groups and stuff. دوستان سوال